Hello, BISC130. This is the beginning of recorded lecture 3.3, and we are about to start on chapter 12 and chapter 13, which are both about genetics. So these are the titles of the chapters from the OpenStax textbook that I'm using: Mendel's Experiments in Heredity and Modern Understandings of Inheritance, kind of wordy. Personally, I think that the way the topics are organized in these two chapters is a little unintuitive. So anyway, what I'm going to do is I'm scrapping this type of organization. I'm lumping these two chapters together, and I'm just calling this genetics. So I apologize if you're trying to follow along closely with the textbook, that this is going to be, maybe a little bit out of order the way the textbook does it. But I think the way that I've taken these topics and just put them in a different order is going to make more sense, is going to be more intuitive, is just going to be easier to understand. So these two chapters lumped together, I'm just calling genetics. Okay, so some terminology to start off with. Chromosomes contain genes. This is a concept I've introduced several times throughout this quarter, but yeah, the chromosomes we have the instructions for making things. The term that I'm introducing, though, is the term genotype. So all of these genes together are what we call the genotype of a cell or an organism. The key terms define genotype as the underlying genetic makeup of an organism. Genotype is describing your genes. It's right there in the name. All the genes you have contribute to your genotype. Now, there's another term here. Most genes, there will be some exceptions. We'll see those in later chapters. Don't worry about that. But for now, most genes encode proteins. They're a blueprint, the instructions for how to build specific proteins. Proteins lead to what is called phenotype. Let me read the key terms definition for phenotype. The key terms say phenotype is the observable traits expressed by an organism. So importantly, this is, what you can see with your eyes. This cat has short hair. This cat has gray and black fur. This cat has a tabby patterning of stripes. So phenotype is stuff that you can actually see. You can't see genotype. You can't look at a plant or a cat or another person and know the set of genes they have, but you can look at a cat or a plant or whatever it is and see these characteristics. So importantly, these are related to one another because genes encode proteins and proteins lead to phenotype. Genotype leads to phenotype. The set of genes that an individual has is going to result in what that individual looks like. Again, there's some gene or genes that are related to having short hair and to having tabby pattern and gray fur. This cat has all these phenotypes because it has all these genes that lead to those phenotypes. Genotype leads to phenotype. Okay, let's talk about inheritance now. So how these things are passed on. What we're talking about is something called Mendelian inheritance. So this is called Mendelian inheritance. It's named after this Austrian monk named Gregor Mendel. We're not really going to talk about his biography or much of his exact experiments and stuff. I'm just bringing him up because that's why this is called Mendelian inheritance, named after Gregor Mendel. It's also worth noting his groundbreaking experiments used the pea plant. It's not the most exciting organism out there, but we're actually going to use the pea plant as an example of how Mendelian inheritance works, because even though it's not very exciting, it actually makes for a very good example to understand basic genetics and Mendelian inheritance. So the first principle here is the idea that there are different versions of genes that can exist. So any given gene can have different versions or different variations. These things are called alleles. Very important term. It's not in the key terms because an allele is just a different version or variation of a given gene. A fantastic example of this, again using pea plants, is the white allele for pea flower color and the purple allele of the pea flower color gene. So these two genes are actually the same gene. This is the flower color gene on this chromosome. Purple and white are not two different genes. They are two alleles of the same gene. This is the purple flower version of the flower color gene. This is the white flower version or allele of the flower color gene. Again, the terminology might be a little, clunky here, but it's very important to note alleles are not different genes. Alleles refer to different versions or different variations of the same gene, as I've summarized here. Now, this seems pretty straightforward. If you've got the purple flower allele, you're going to have purple flowers. If you have the white color allele, you're going to have white flowers. But this gets a little complicated because pea plants, just like us, are diploid. Diploid organisms have two copies of each chromosome. You may remember this figure from an earlier chapter when I first defined diploid. We've got two copies of every single chromosome, which means for pea plants, they're going to have two copies of this chromosome, which means they're going to have two copies of the flower color gene. So diploid organisms have two copies of each chromosome and two copies of each gene. Now, there are several possibilities here for what an individual can have, or a pea plant can have, if we're continuing to use them as an example. If this pea plant, or whatever, has two copies of the same allele, the term for this is homozygous. So if there's a pea plant out there on one of its chromosomes, it has the white flower allele, and on the other chromosome, because it's got two, also has the allele for white flowers, that's homozygous, two of the same thing. Here's another way to have homozygous. If you're a pea plant and you've got a purple flower allele on one of your chromosomes and also the purple flower allele on your other copy of that chromosome, that's also homozygous. Two of the same thing means homozygous. What phenotype are they going to have? Well, it's pretty obvious. If you've got two of the white flower allele, you're going to have white flowers. And if you have two of the purple flower allele, you're going to have purple flowers. This much is pretty easy. Let me summarize this here. So these homozygous individuals will have the phenotype of the allele version they have. Again, they're either going to have purple flowers or white flowers, depending on which allele they have two of. But what if you have two different alleles? So an individual can have two different alleles, and the term for this is heterozygous. And what's the phenotype going to be? Well, yeah, that's a bit of a question mark here. If you've got a purple allele and a white allele, what is that going to lead to? Again, genotype leads to phenotype. What phenotype is going to come out of having one of each? Well, the answer to this question actually depends on what these genes are doing. So we're going to look deep at this exact example, but again, you'll see in my summary that we're not memorizing all of these details. This is just to kind of walk through this particular example to understand, in this way, for flower color, how genotype leads to phenotype. So, okay. Background. Flowers look purple because they have a pigment molecule called anthocyanin. Yeah, this is not a pea plant, this is a cauliflower, but yeah, it's purple. There are lots of things in nature that are purple, vegetables in nature that are purple, plants in nature that are purple. They're all purple because they're making this class of pigment molecule called anthocyanins. Pigments should be a familiar concept to you. We saw these in the photosynthesis chapter. Here were, green pigments reflecting green to your eyes and absorbing everything else. These things look purple to your eyes because this pigment reflects purple back to your eyes and absorbs all the other wavelengths of visible light. So that's why these things are purple, but where do these anthocyanin pigment molecules come from? Well, we get anthocyanin. I keep saying we, pea plants. Pretend you're a pea plant for the next 10 minutes. We get these anthocyanins from, one of these. So do not memorize any of this, but the basic structure of what we're looking at here should look familiar to you. This is a metabolic pathway, kind of like glycolysis was a metabolic pathway. Specifically, this is one of those assembly lines. This is an example of anabolism, an anabolic pathway. Through a series of intermediates, every single one with its own enzyme here, these anthocyanin pigment molecules can be created by, pretty simple starting materials that every cell is going to have. So, all right. What does this have to do with the purple allele, with the purple gene? Remember, genes encode proteins. This purple flower allele in the DNA is the blueprint, the instructions for building this protein. What does this protein do? Well, this protein doesn't build anthocyanins. Remember, there's like 1, 2, 3, 4, 5. There's a bunch of proteins in this pathway that contribute to building anthocyanins. This protein acts as a switch for the entire pathway. We'll talk more about switches and gene regulation and the details of how things like this do their job in a later chapter, but for now, just know that this protein turns on the pathway. So if you have a purple flower allele, that means you're making this switch protein, you're turning the pathway on, and you're building anthocyanins, and your flowers are going to look purple. In a homozygous purple flower, you've got 2 of this allele. So you're making double the copies of this protein. Of course, the pathway is going to be switched on, and the flowers are going to look purple. This is what we talked about earlier. Homozygous purple is going to have purple flowers, but now we know why. The genes make the proteins turn on the pathway builds pigments, the pigments make it look purple. So, what about the other allele? This is what the purple version of this gene does. What about the white version? Well, as it turns out, the white allele for this flower color gene encodes a defective protein. It's sort of like a broken blueprint that doesn't build what it's supposed to build. It doesn't really build anything at all. So this white allele it encodes a protein that doesn't do anything. So it's not going to switch on the pathway. Without the pathway switched on, none of these chemical reactions are going to happen. You're not going to build anthocyanins, and so the flowers are not going to look purple. They're going to look white, which is sort of like the default color for most things. If you're homozygous for this white allele, you could imagine what happens. You've got 2 defective genes making double the defective copies. It's still not going to work. It's going to be turned off. It's not switched on. None of these anthocyanins are going to get made. Flowers are going to look white. Again, this is what we said before. If you are homozygous white, flowers are going to be white, but now we understand the order of events, why this happens. Defective proteins, pathway not switched on, no pathway, no purple, white instead. Now, I brought all of this up so that we could understand what happens in the heterozygous situation. So for this gene, for these alleles, what happens in a heterozygous situation? Well, for heterozygous, we have a purple flower allele. This allele encodes this switch protein, which could switch on the pathway and make purple. But we also have the white flower allele. The white flower allele encodes a non-functional protein that doesn't do anything. This protein that doesn't do anything, though, doesn't get in the way of a functional copy. It just doesn't do anything on its own. So if you have a working copy of this switch and a dud, the switch is going to get turned on. It's going to get turned on just as much as it was turned on with 2 purple alleles. and you're going to have activation of this pathway. Cells are going to build anthocyanins. Flowers are going to look purple. They're going to look just as purple as they did with homozygous purple because, either the pathway is on or it's not on. It can't be like more on because there are more of this protein. It's just as on as it was in either of these circumstances. So whether you're homozygous purple or heterozygous, the phenotype is going to be the same, purple flowers. And again, the only reason we know that this is going to be purple is because we understand this pathway. Again, it's important to appreciate that alleles, these genes, encode proteins, and the proteins do stuff, and the stuff that they do results in these phenotypes. So there are a couple of terms here, dominant and recessive. So in the relationship between the purple flower allele and the white flower allele, we say that the purple allele is dominant and the white allele is recessive. We say that because of this relationship. When you put the two of them together, the phenotype is purple. White just kind of disappears here. Purple is sort of stronger than white in this, matchup between the two of them. That's why we declare that purple is dominant and white is recessive. And again, we know this because of all the biochemistry of what happens. So again, this is a lot of information, but what's my take-home message from this? So here's my summary of all of this. For any given gene, whether it's flower color or whatever, one allele is dominant and the other allele is recessive. If we want to look specifically at pea flowers, purple is the dominant allele, white is the recessive allele, and we know why because of this. Heterozygous individuals will have the phenotype of the dominant allele. So here's a heterozygous individual. It's got one of each. What is its phenotype going to be? Its phenotype is going to correspond to whichever of these alleles is the dominant allele. In this case, purple is dominant, so the phenotype of a heterozygous individual is going to be purple. Example with pea plants, because purple is dominant, heterozygous individuals are going to have purple flowers. Importantly, there is no light purple. There is no in-betweensies. There is no blending here. The heterozygous' purple is just as purple as the purple of the homozygous. Again, you don't take the average or anything like that. It's either purple or white. Two options here. The heterozygous individual looks exactly like the homozygous purple individual. Now we have some terminology. We've even more terminology. There are three possible genotypes: homozygous dominant, homozygous recessive, and heterozygous. Let me walk through these, but again, these should be familiar. This is just, we already defined homozygous. Now we're just throwing dominant and recessive into the mix. A homozygous dominant individual is an individual that has two of the same thing. That's what homozygous means. And the thing that they have two of is the dominant allele. Homozygous recessive individuals have two of the same thing. That's what homozygous means. But the thing that they have two of is the recessive allele. Heterozygous individuals have one of each. Importantly, and I will try to trick you on a multiple choice test. Well, not really trick you, but whatever. There's no such thing as heterozygous dominant or heterozygous recessive. Just even thinking about that should just not make any sense at all. Heterozygous means you have one dominant and one recessive. So heterozygous dominant just doesn't make any sense. Heterozygous recessive just doesn't make any sense. It's just heterozygous, one of each. So when we're talking about genotypes of a specific gene, these are our only three options. Homozygous dominant, two of the dominant. Homozygous recessive, two of the recessive. Or heterozygous, one of each. And importantly, all these genotypes lead to a specific phenotype. The homozygous dominant genotype leads to the dominant phenotype, purple in this case. Homozygous recessive genotype leads to white flowers, whatever the recessive allele phenotype is. And the heterozygous genotype leads to the dominant phenotype because, we got two of them together. The dominant is stronger than the recessive. You end up with the dominant phenotype. Here's my summary slide. 3 possible genotypes. Homozygous dominant has a dominant phenotype. Homozygous recessive has a recessive phenotype. And heterozygous has the dominant phenotype. I'm going to use these terms, homozygous dominant, homozygous recessive, heterozygous. I'm going to use these throughout this genetics chapter, so you should really become familiar with these 3 terms used to describe genotype. Let's turn our attention now to inheritance, to how these traits and these alleles and genes are passed from parents to offspring. So if we're talking about, here we go. This should look familiar. Use this in an earlier chapter talking about reproduction. So both parents, again, whether they're humans here or whether they're pea plants, both parents are going to be diploid. But when it comes time to making offspring, to making sperm or eggs, as the case may be, only one copy of each chromosome goes into the sperm. Only one copy of each chromosome goes into the egg. So parents are both diploid, but they only pass one of each chromosome to their offspring. Back in the chapter when I used this slide, I introduced this analogy of diploid, meaning having 2 slightly different chairs and 2 slightly different tables and 2 slightly different nightstands and so on. So yeah, when it comes time, each parent is diploid, but when it comes time to making eggs or sperm, they make haploid cells. They make haploid rooms. Only one of each piece of furniture gets passed on to the offspring. And of course, which one goes is chosen randomly. We talked about all this in the meiosis chapter. So which version of each chromosome that they put into any given egg or sperm is chosen randomly. This results in many possible outcomes. And again, this increases genetic diversity. That's really good for having diverse offspring. Now, there are many possible outcomes here, but we can predict what these outcomes could be. The way that we predict the outcomes is through things called Punnett squares. These are diagrams that are used to predict the possible genetic outcomes of offspring from parents. This is usually done one gene at a time. You could do bigger, more complicated ones that track multiple genes, but everything we're going to do in this class is just looking one gene at a time. What do the parents have? What combinations could the offspring have? So there are some rules to building Punnett squares. And we will draw some of these. I'll show these in just a couple of minutes, but let's outline the rules first. So again, we're tracking one trait, one gene at a time. The trait or the gene is assigned a letter. So let's, again, keep looking at flower color. By convention, the letter that we choose is usually the letter associated with the dominant phenotype. So for flower color, as we've seen earlier, the dominant phenotype is purple. So by convention, the letter we would choose is P. We will see in just a minute why this is a bad idea, but again, by convention, usually associated with the dominant phenotype. Individuals are diploid, so they're going to have 2 of that letter. So, the parent, each one of these parents is going to have a P and another P. How do we distinguish between dominant and recessive alleles? Again, P doesn't refer to any specific phenotype. P is just talking about flower color. What if there's a white allele? Do we use W? No, we don't use W. Never use W. We have to distinguish between dominant and recessive by doing capital and lowercase. So the letter is capitalized if it is the dominant allele and lowercase if it's the recessive allele. Again, they're both P because they're both the same gene. Now you can see why P is a bad choice. So dominant, purple flowers, capital P, recessive, white flowers, lowercase p. These look too close to one another. These are easy to mix up with one another. I don't recommend ever, even if convention dictates you should use a letter associated with the dominant phenotype, I don't recommend ever using letters where the lowercase and the uppercase are easy to mix up with one another. I would use A. A is an easy one, whatever font you're using. Capital A and lowercase a are very easy to distinguish. So, just by default, it doesn't matter what letter you use, really. A is a good choice. So, knowing that dominant is capital A and recessive is lowercase a, this gives us a very neat shorthand for describing those three genotypes that we defined earlier. So homozygous dominant, having two of the same thing, and it's dominant that you have two of, can very easily be written out as capital A, capital A. Homozygous recessive, having two of the same thing, and it's recessive you have two of, is just lowercase a, very succinct. And of course, heterozygous means one of each, capital A, lowercase a. I guess you could do lowercase a, capital A. The order doesn't technically matter. By convention, usually the heterozygous is written like this, with the capital letter and then the lowercase letter. So yeah, these are important ways to sort of abbreviate these three genotypes using these conventions of capital and lowercase. So, where's the actual square? This was just backstory. So, to do a Punnett square, you start by drawing a square, and then drawing a couple of lines through it to create a 2x2 grid. So, not really a Punnett square, more of a Punnett grid, but, it is what it is. We are going to write out the genotypes of the parents on the top of this grid and on the left side of this grid. So we could do whatever parents we want. As an example here, let's say one of the parents is homozygous dominant, and let's say the other parent is homozygous recessive. Let's put some visuals in here just to remind us what these things mean. Homozygous dominant means two of the purple allele, and homozygous recessive means two of that white allele. Okay, so how are we going to fill this out? Well, let's use our shorthand. Homozygous dominant means capital A, capital A. And we're going to put it up here just like this, with one of these lined up with these two squares and the other letter lined up with these two. Homozygous recessive means lowercase letter, and we're going to put them over here on the left, lined up with these squares just like this. So this is just describing the parents. The letters up here are describing one parent. The letters over here are describing the other parent. This is supposed to look at the offspring. The offspring are going to be in here. These four boxes are going to describe what the offspring could be. So to fill out the boxes in this grid, we start by taking this letter and dragging it down to fill out this box. It's a capital A, so we're going to fill in this box with a capital A. We're going to drag it down further to the other box, fill out this one with another capital A. Now, we're going to do the same thing with this A. We're going to drag it down, fill out this box, it gets a capital A. And this box gets another capital A. So we filled out one of the parents. Now we have to do the same thing for the parent over here. The lowercase a here gets pulled to the right and fills out this box. It already had a capital A, now we're adding a lowercase a. Going to drag it over to this box gets a lowercase a as well. Now, we move down here, pull this lowercase a into this box, it gets a lowercase a, and pull it even further to this box, now it gets a lowercase a as well. So, we've finished filling out this square, but what does it mean? Well, each one of these four boxes represents 25% of the offspring. Because there are four of these, 25%, 25%, 25%, 25%, it totals up to 100%. Again, this is predicting what the offspring could be. Each of the four boxes represents a 25% probability of possible offspring. Now, You might be wondering how I'm going to grade this on a multiple choice test. Yes, all my tests are multiple choice. I'm not going to, like hand grade your Punnett squares, but what I could do is I could ask you a question like this. In a genetic cross between a homozygous dominant parent and a homozygous recessive parent, what percentage of the offspring will be heterozygous? So this is heterozygous, this is heterozygous, this is heterozygous, this is heterozygous. The answer, 25% plus 25% plus 25% plus 25%. The answer is 100%. 100% of the offspring will be heterozygous because each box is 25%. Here's another question. I could say, in a cross between homozygous dominant and homozygous recessive, what percentage of the offspring will be homozygous dominant? The answer to that question would be 0. None of these offspring are homozygous dominant. So maybe you see where I'm going here. The questions will always be what percentage of the offspring will be, so-and-so, and you'll have to be able to draw and fill out this Punnett square and, add up 25, 25, 25, 25 to get whatever the percentage is. Let's do another one. More practice here. So let's do a slightly more interesting cross. Let's cross a heterozygous parent with another heterozygous parent. Visuals, heterozygous means one of each. Heterozygous means one of each. Capital A and lowercase a. Again, it doesn't technically matter whether you put the uppercase on the left or the right or whatever, but by convention, we always put them in this order, with the uppercase, the dominant allele on the left, and the lowercase on the right. Uppercase on the top, lowercase on the bottom. So yeah, we filled this out just like we did before. Drag this allele down, drag it down further, drag this allele down, drag it down further. We're halfway done. Now we're going to drag this allele to the right, drag it further to the right to fill out this box. Again, we had a lowercase a here. We added an uppercase a. By convention, we always just put them in this order with the uppercase on the left and the lowercase on the right. Take this lowercase, drag it over to fill out this square. Take this lowercase, drag it over to fill out this square, and there we go. Again, these are the questions I could ask. So yeah, you do a cross between two heterozygous parents. What percentage of the offspring will be heterozygous? The answer is 50%. This box is heterozygous, this box is heterozygous, 25 plus 25, 50%. What percentage will be homozygous recessive? The answer is 25%. This one box is homozygous recessive. Ooh, I could get tricky. I could ask, what percentage of the offspring will be purple? What percentage of the offspring will have the dominant phenotype? Here we have to think back. This is going to be purple. Homozygous dominant has the dominant phenotype. This is going to be purple. Heterozygous has the dominant phenotype. Heterozygous has the dominant phenotype. This is going to be purple as well. But this is going to have white flowers. Homozygous recessive does not have the dominant phenotype. Homozygous recessive has the recessive phenotype. So if I asked you what percentage of the offspring are going to have purple flowers, the answer is 75%. 25 plus 25 plus 25. So again, these are the sorts of questions that I could ask on an exam. Let's do one more. And for this one, I'm not going to do this one piece at a time. For this one, if you really want some practice, if you got this down already, that's great. But if you really want some practice, pause this video, draw this out yourself on a piece of paper, fill out the individual letters, and then hit play on this video again. I'll give you the answer. But yeah, if you want some practice, pause and do this on your own. If you're coming back to this now, your Punnett square should look like this. So once again, if I asked a test question, you're doing a cross between a heterozygous parent and a homozygous recessive parent, what percentage of the offspring will be homozygous recessive? The answer, 50%. Homozygous recessive, 25 plus 25, 50%. What percentage of the offspring will have the dominant phenotype? 50%. Heterozygous individuals have the dominant phenotype. Homozygous recessive individuals have the recessive phenotype. So this is going to have the dominant phenotype. This is going to have the dominant phenotype. This 50% is going to have the recessive phenotype. So again, a lot of practice here, but those are the sorts of questions that I could ask. One more thing about this. So when I write a test question, I'm going to say a cross between a heterozygous parent and a homozygous recessive parent. How do you know which one to put on top and which one to put on the left? Well, it doesn't matter. So if you put the heterozygous on top and the homozygous recessive on the left, your square would look like this. If you put the homozygous recessive on top and the heterozygous on the left, your square would look like this. So technically, the squares do look different depending on, which parent you put on the left and which parent you put on the top. But all the numbers, all the results are exactly the same. What percentage are going to be homozygous recessive? It's still 50%. What percentage are going to have the dominant phenotype? It's still 50%. So do not worry about which one goes on the left and which one goes on the top. It does not matter which parent goes on the left and which parent goes on the top. You will get the same results either way. Now, we've been talking a lot about pea plants because, again, they're a convenient example and there's some historic reasons for this. But I do want to point out, before you get too bored with all of this, that there are a lot of human traits and human diseases that follow everything that we've talked about. Again, don't memorize this table, but it is an interesting one to look at. Huntington's disease is a dominant trait. The allele for Huntington's is dominant. The allele for, normal, brain protein, whatever, is recessive. If you've got one Huntington's allele, you will have Huntington's disease because it's dominant. These are recessive things. Cystic fibrosis is recessive. If you've got a cystic fibrosis allele, if you've got just one of them, you're going to be fine. If you've got two of them, you're going to have cystic fibrosis. So again, just pea plants can be kind of boring. I just wanted to point out in this single statement, many human traits and human diseases follow what we've talked about, follow Mendelian inheritance patterns. We use the same terminology of homozygous dominant, homozygous recessive, and things like that. You can make Punnett squares to track cystic fibrosis or Huntington's disease or phenylketonuria or any of this stuff. There's a lot of actually interesting human things that follow everything that we've talked about so far. Okay, we are not done with the genetics chapter. This is the genetics chapter. It's a couple that have been lumped together. We are not done talking about genetics, but that is enough for this recorded lecture. We will pick back up and do more genetics in the next one, but this is the end of recorded lecture 3.3.